Hi. For lecture 14, I'm going to be talking about how we're taking genomics and applying it in the context of human health. And hopefully by the end of this lecture, you'll have a better ability to recognize the broad causes of disease, understand the variability of phenotypic responses to mutations, be better able to be familiar with the methods and results of genome-wide association studies, or GWASs, have an understanding of the human microbiome and its role in health and disease, know about the role of genomics in etiology, diagnosis, and treatment of cancer, and recognize that cancer is a disease of genome instability. So I think it's really important when we think about genomics in human health that we first start off and take into consideration the concepts of precision medicine and personalized medicine. So these are terms that you'll often hear used in a modern medical environment and also in professional and popular literature. And they don't exactly mean the same thing. So precision medicine refers to a medical model that proposes the customization of healthcare, where decisions and practices and products are tailored to individual patients. And one of the big applications we see here is in diagnostic testing, right? So diagnostic testing is used to select for appropriate and optimal therapies based on the content of a patient's genetic information or some other kind of molecular cellular analysis. And these include tools related to molecular diagnostics, imaging, and analytics. Personalized medicine is targeting more of a medical procedure that separates patients into different groups. So where we have medical decisions and practices and interventions and even products that are tailored to individual patients based on their predicted response or risk of disease. So there's a little bit of a distinction between these of these different groups, where we have precision medicine, which is trying to give us the best customized healthcare model possible, and personalized medicine, where we're really trying to identify the specific practices and interventions that can be tailored to specific individual patients. And genomics has the capacity, in other omic fields, transcriptomics, proteomics, metabolomics, all these omic fields, have the ability to really impact both of these two areas. And of course, when we're talking about human health, we are often going to be, and especially in the context of thinking about genomics, we're really going to be focused in on, the diseases that we see associated with genetic mutations, genetic variants, specific genetic variants. And this, of course, relates directly to some of the work that we're doing in class on the project with our clinical variant work. And so it's just important to note that some diseases are associated with mutations in specific genes, right? And this is what let us understand that there's an underlying genetic association or mechanism that's associated with specific types of diseases. How we tend to identify whether or not a specific disease phenotype is associated with a genetic factor typically comes from us looking at the genotype-phenotype correlation. So assessing when we see a genetic change, do we also see another change in the phenotype that follows that genotypic change? But one of the things that comes into this is that we need to move beyond these correlations. We actually, to really be able to treat and understand how we can use genetics in a disease context, we definitely want to be able to understand how mutation leads to that specific disease. So showing a correlation is informative and useful, but what we really need to know is what are the causative agents in that process. And this will help us get a better understanding. One of the terms that we can use to better understand this is penetrance. So when we talk about penetrance, what we're talking about is the fraction of individuals carrying an allele for a condition who actually manifest it. right? And so by understanding sort of the difference between correlation and causation, if a gene was the causative factor of a specific disease phenotype, then we would generally see that allele always be associated with that disease. But sometimes we don't see that to be the case. Sometimes penetrance is often, they're typically not 100%. And so it's important to understand that there can be multiple factors, or that the thing that we're looking at, the genetic marker, may not actually be the cause of the disease we're looking at. It's also important when we consider what is the effect of a mutation on an actual disease, is to think about the concept of variable expressivity. Variable expressivity basically refers to, among those who carry a specific allele, what is the severity of the condition. So when we see individuals that have a specific mutation related to, say, something like cystic fibrosis, are they all experiencing cystic fibrosis in a similar fashion, or is there a lot of variation among their symptoms and responses? And by understanding these sort of context dependent phenotypes, and what we would often think of as the outcome of a specific genetic variant, we can better also understand the role and the importance of a specific mutation in the actual disease phenotype that we're interested in. Classically, how we've looked at these sort of things is we've looked at it from a pretty Mendelian classical perspective. So, if a disease can be attributed to some sort of genetic mutation, we're going to look to identify what are the relationships between parents and offspring. Often we'll go for the big, easy targets. We'll say, okay, if we know that there's a protein involved, can we target that protein for therapy? If we know it's a gene involved, can we identify sufferers and carriers? This is a classic way that we've thought about examining these kinds of problems. But the issue is that was really easy for us to do for a certain set of diseases, but most diseases do not show the kind of simple patterns of inheritance that make them easy to track in this fashion. So it's become it's a much more difficult process than what we think. Many genes, many diseases are multigenic or multiallelic, and are polygenic, and so it makes it really difficult to identify all of the contributing factors. They're also context dependent, so environment, development, a lot of other factors can contribute to the manifestation of specific disease phenotypes. Sort of the classic example of genetic diseases, we can go back and we can look at something like sickle cell anemia. Back in the late 40s, Linus Pauling was able to show that that there were electrical charge differences between different types of hemoglobin, specifically these types of hemoglobin that were exhibiting these sickle cell phenotypes. And eventually, these differences were tracked to mutations in residue 6 of the beta chain, to a nonpolar residue, and what that mutation caused, what this non-synonymous mutation caused, was a sort of sticky path that formed, which leads to this sickle cell phenotype. This causes polymers to form in the deoxy form of venous blood. And what happens is that erythrocytes they lose their sort of deformability, their ability to squeeze through small passages and flexibility, and they often block capillaries. This then can lead to all kinds of hypoxia issues in tissues, and can cause things like pain, anemia, jaundice, and other serious outcomes. This is kind of one of the most simple cases where you have sort of one individual mutation leading to a specific amino acid change, and that causes a big phenotypic change, which then underlies a very well-known and identifiable disease phenotype. There are a range of other blood disorders, things like thalassemias, where we see that you have less hemoglobin and fewer blood cells in your body than normal. Right. There are kind of two main types. There's alpha thalassemias, where we see defective or deleted. We see the lack of either a defective or deleted hemoglobin gene. Basically, we have 4 genes for alpha chains in adult hemoglobin, and the disease happens when there's less than 2 functional copies of these genes. This presents differently depending on how many genes are still functionally, but it leads to general instability in transcripts, in some of the transcripts, which can cause this fewer red blood cell phenotype. Additionally, we have beta thalassemias, when these are usually not large deletions, right? So in other words, we not having a deletion or defective hemoglobin gene. This is usually the result of point mutations, which result in either missense mutations, where we have amino acid substitutions, or nonsense mutations, where we have stop codons added, and a premature termination or truncation of the protein, or say a mutation in a splice site, or a mutation in a regulatory region, or some sort of deletion between codon termination and the intergenic region. So this often will result in some kind of fusion protein, where protein sections are fused together where they shouldn't be. So in the previous case, we had sort of this deletion, which is the genetic mechanism underlying the phenotype. And in this case, it's more point-based mutations, a single mutation, which is leading to this phenotype. So even though phenotypically they may look very similar to each other, the mechanisms are actually different. So we can start to see how the type of genetics that underlie these systems is actually a little more complex than what we might think. Moving along in the same space, we can come up to some other genetic disorders like phenylketonuria. And this is a disease which is a deficiency in the metabolic enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine. And what can happen in this case is if you have a deficient or an unfunctional phenylalanine hydroxylase, you get a buildup of phenylalanine. Right? And this can lead to all sorts of developmental defects. This trait is generally identified as being an autosomal trait, so not a sex-linked trait. It's a recessive trait, however. So we have shown that there's essentially mutations in all 13 exons and flanking regions of this gene. So there's a lot of different spots in one gene that could be related to the sort of lack of function in this specific gene. One thing about phenylketonuria is there actually are some ways to treat it, right? So you can treat it through things like giving people a low phenylalanine diet, although this is often difficult to do because it's not all that tasty and it's difficult to manage from a nutritional perspective. There are also some enzyme replacement therapies that are in place now where they actually will use the phenylalanine ammonium lysase and use that to convert phenylalanine to a more usable process. And so there are some ways that people have been trying to address these protein deficiencies. So in this case, you can see that there are some, again, there are different types. Now we see that their entire gene, there's a whole range of mutations. Again, we had the easiest case to start off with sickle cell anemia, but now we can see that, there can be a whole different set of possible mutations in a whole different parts of these genes that can drive these types of disease phenotypes. Alzheimer's disease is another example where we have a strong we start to get a lot of, we can see evidence for there being a role of genetics in this process. Of course, Alzheimer's is the loss of cognitive function that we see often in people who are elderly in our population. It's typically associated with the formation of amyloid plaques that lead to these neofibrillary tangles of tau proteins. And about 50% of people age 85 or older suffer from it. There is a risk of late onset Alzheimer's correlated with specific SNPs in the apolipoprotein E, which removes cholesterol from the blood. And one of the things that's interesting in this case is that, if you have a specific allele in this gene, you have an increased risk, almost 3 times an increased risk for developing Alzheimer's. But if you have a different set of alleles, allele set, you would actually have a decreased risk. And I think what's important to see here is that when we think about, well, what are these common alleles? What we're actually looking at is a combination of different SNP variants. So the allele is not just a single locus allele, it's a 2 locus allele. So we can see that, for instance, apolipoprotein allele 4, right, has a C in one position and a C in another position, right? And this is your increased risk allele. Whereas your apolipoprotein allele E2, right, has a T in that first position and a T in that second position. What this can show you is that while this is just one gene that's affecting this process, we can have multiple loci within a gene contributing to this process. So now it becomes a multi-locus genetic process underlying a disease phenotype. And I think it's important to note that, as we go through and identify these different types of phenotypes, that, they start to expand beyond just one gene, right? So if we think about something like schizophrenia or some other whole suite of other different diseases, we often think that these are maybe under multigenic or polygenic control. So it could be even more than one gene spread, multiple genes spread across multiple chromosomes. You can start to see how complex this thing can get. So how do we investigate these types of genetic relationships? The classical way is to create a gene map using linkage patterns of heredity in affected families to localize the effects of genes in a region. And to do this, one of the first steps is often to build a pedigree of individuals. We'll start with a proband, say like a person affected by this disease, and then we'll create their pedigree around them and identify the status of individuals in that pedigree with regards to whether or not they were affected or not affected by the disease. This helps us identify, which individuals may be carriers or not carriers of the disease, based upon which ones are affected, not affected, and what generation they were. Then we'll go through and sequence these individuals and try to find regions that are associated with individuals that have this disease. And once we're able to do that, we can start to pinpoint, which genetic features are actually responsible for the manifestation of the disease phenotype. Another way to do this is to take more of a population level perspective or population level study, and is to generate what's known as a genome-wide association study. So in this case, we would identify regions in the genome for which sequence differences correlate with a particular phenotype. There are sort of two sets or subjects. There are two sets of subjects that you would take in this. There's usually a disease versus a non-disease group, right? And you're going to do a comparison between the two. Usually when you do this, you end up screening each subject for a large number of scripts, anywhere between 500,000 to 2 million or more. And then what you're looking for is to correlate specific nucleotide SNPs with the presence of a trait or with the presence of a specific trait to identify a region of interest. So we're looking to see if a specific allelic call is associated with a specific genetic outcome, or in this case, a phenotype for a disease. This is a process which is most powerful when the alleles are of large effect. And the hope is that you're going to hit a haplotypic block that contains a gene involved in that trait. So, we're only looking at 500,000 to 2 million actual locations in the genome. Of course, the human genome is 3.3 billion bases long. We're not going to be able to sample every single base with this type of methodology. So what you're often looking for is you're looking for the fact that you might identify a haplotype block, right? So you might have a genotype SNP in this block, and a disease risk SNP may be in that block as well. But just because you see a genotype SNP associated with a disease outcome does not mean that this genetic variant specifically is the contributor to that disease. It just means that something in that block might be. So that's a really important point to recognize. When we're looking at the types of different things that you can identify with GWAS tests, we can see that there's sort of different levels of efficacy based upon what is the allele frequency of the genetic of the allele that's causing or associated with the disease phenotype in the population, and the effect size. So how strong a relationship or how strong the relationship is between that variant and the actual phenotypic expression or observed phenotype of that disease. So for instance, common variants can have really large effects, can have a big influence in Alzheimer's, right? So they're common, so it's easy to see allele frequency differences. The effect size is big, so maybe it's a little bit easier to see some of those things. Typically, actually, it's things in this middle band that are the kind of the most easiest to detect, where we have this sort of intersection between allele frequency and effect size. So where we see highly penetrant Mendelian mutations like in cystic fibrosis, or where we have less common variants but with moderate effects like Crohn's disease, or even where we have common variants but they have small effects because they're commonly observed, they're easy to show these associations. So things like prostate cancer are easily observed. Where things are rare, it's often and have small effect, that's often really hard to see. So when we have a lot of rare variants with small effects, maybe all multi-genic examples making lots of different contributions, it's really hard to observe these sorts of things. So that's something to keep in mind. This is what a Manhattan plot looks like. This is a classic way to display GWAS information, and I think it's really important that you understand what we're looking at here. So what we have here is a case where we have each of the chromosomes lined up, and then we have these positions along the chromosomes, and we're looking for differences, evidence that there are significant differences between our disease versus non-disease groups. So in this case, this was a GWAS looking for genetic variants that were going to be associated with the ongoing expression of fetal hemoglobin. And so hemoglobin, there are changes in hemoglobin expression through time. One of the ways, when we talk about sickle cell anemia, one of the ways to ameliorate that is that's typically a mutation that we would find in the adult version of hemoglobin, right? So one of the ways to ameliorate it has been hypothesized as to, well, why don't we just keep on expressing fetal hemoglobin rather than trying to fix it a different way, right? So is there a way that we can maintain the expression of fetal hemoglobin, which may not have mutations, in a way to replace the deficit of adult hemoglobin? And so, what we're able to see in this case is that we can identify a few specific regions along these chromosomes that are significantly associated with this disease. And what we can find is here we have, this is a log scale 10, so 0.1, 0.01, 0.001, etc. of p-values, right? And so the p-values indicates, it's significant association with the disease. And so the higher the p-value, the more the more certain we are that that association is real. Or sorry, the smaller the p-value, the more certain we are that association is real. And so we have these lines here, and these red lines can indicate corrections for multiple, either threshold values that we would want to put on there, or corrections for multiple comparisons, to sort of put some additional thresholds of stringency on whether or not we think an actual SNP is significant or not. So in this case, if we were to take the 1 to the minus 7 p-value is our stringency threshold, we can identify 1, 2, 3 specific regions where there are really significant associations with the expression of fetal hemoglobin in adults. And it was interesting because when they actually did this, they found that one of these mutations was actually in the area of a gene known as BCL11A, which is a zinc finger protein, which is known to control fetal hemoglobin expression, right? And there have been other works to show that, like, when this gene is inactive, it leads to neonatal lethality, and that when you have examples, like in cases like this, where it's where you might have a mutation in BCL11A, and it is inactivated, we actually see an increase in fetal hemoglobin. So its expression seems to be important in controlling the expression of fetal hemoglobin. So maybe this is a good target for figuring out ways to manipulate the expression of fetal hemoglobin in humans. So those are some of the ways we can think about, the human genome and its relationship to disease, how we can start to take a genomics perspective and see how, maybe if we can understand the genetics of somebody, we can start to better understand, various types of treatments. Should we look for, is the kind of treatment we're looking at going to be related to an enzyme therapy? Is it going to be related to something that's going to have to do with changing one's diet to better treat something, right? Is it going to be related to thinking about genetic interventions that we might take to either increase or repair a damaged gene? These are the kinds of things that people are thinking, and there are all kinds of ways that we are doing actual genetic manipulations right now in humans using tools like CRISPR-Cas9 or adenoviruses to, insert genes into various organs to produce different enzymatic products, right? And so there are ways that we could think about approaching these and addressing the genetic deficits directly. But it's not only our own genome which is interesting. The human body is a system that includes a lot of other organisms beside just those that are our DNA. So there are bacteria, archaea, viruses, and fungi that are all living in symbiosis with us. In fact, there's about 30 trillion human cells. There's about 30 trillion cells in a human, but we house about another 39 trillion cells of bacteria. So there's about 150 times as many genes in this microbiome, which is what we refer to it, as there is in the human genome. So there's actually more genes in the microbiome than there is in the human genome. And it makes you kind of question of like, well, what is an organism in a species if, so much of our genetic material and so much of what we're doing is actually made up of organisms from other groups, these microbial organisms. So a while, it took us, we've known that these bacteria in our gut and in other parts of our body, and there have been a push for us to sort of examine what they do and how they work. So classical methods for this involved us sort of looking at each pathogen in and of itself. So we would, take a sample of something, we'd clone those, we'd culture and clone those pathogens, and try to identify, what we were looking at in each of these different cases. But the problem is that most of the microbes that we're interested in aren't easily cultured. And then it's also really difficult to observe what are the actual interactions between the microbe and host, because we're doing it in a lab setting and not in vivo. And these include all kinds of interactions could include all kinds of ways that microbes and hosts may be contributing to gene expression modulation of each other. They could be shaping the immune system, and they are both have a certain amount of robustness to them, to these systems, and also variability that is difficult to track when you're looking at one organism or one player at a time. Right? We're talking about a system of a lot of different types of bacteria all interacting with each other. So the classical technique didn't really work. So there have been some more modern techniques that we've tried to integrate. These have included things like amplicon sequencing, where we take a DNA sample from a microbial sample, say from a skin swab or a stool sample, and we amplify the 16S ribosomal rRNA sequences in these microbial communities, or maybe something like the internal transcribed spacer gene, which is we can refer to as ITS, so 16S or ITS. And then we use these to identify what are the species-specific signatures that are in these components of the microbiome. So we take stool, we isolate DNA from it, we amplify the 16S, and we sequence it, and we say, okay, what do I see? Do I see sequences associated with different, like these, what species of bacteria do I see associated with these sequences? Can I identify these sequences with what's known about other species of bacteria? And I can kind of work out the proportions of these different bacteria based upon that, right? So this is one way that I can do this. ITS is typically used for looking at fungal targets. So in this way, I can look and see what species are present in a sample. I can look and see, depending upon how good I am at it, I can also see at their relative abundances. So this is one of the standard ways that we would do sort of this microbiome investigation. What is becoming a more commonly applied approach, though, is to take a more whole metagenome sequencing approach, is where we instead of amplifying a specific sequence from a DNA sample, which can lead to some biases and misrepresentations in some ways, we instead do whole metagenome sequencing. And this allows us to assemble genomes from everything that's in a DNA sample. And it also lets us identify not only just 16S sequences, but also looking at other kinds of functional genetic elements. So we can look and see what genes are most prevalent, how much variation we see in them. How does this relate to the functions that we think something might be doing? This is also really important when we start thinking about how this translates into human health context, as there is an analog to the genome-wide association study with the metagenome-wide association study. And in this case, what we're looking at is the correlation between community makeup and disease. So we can look at the frequency of different bacteria, but we could also look at the frequency of different bacterial genes and disease. And the number of copies of these genes and their abundances, we can then look to correlate we think are well correlated with sort of the frequency of these organisms. So we can kind of look and see which bacteria may be mostly contributing to different types of or to different types of disease states or resilience to those disease states. So right now there are broad initiatives to sort of map this diversity within the human microbiome, looking at things like oral microbiome, nose microbiome, vaginal microbiome, intestinal microbiome, all these kinds of things. And we see different species active in different spaces. And by understanding sort of what the relationships we expect to see, we can start to understand what a healthy microbiome might look like and what an unhealthy microbiome might look like. And some of the ways that we know that microbiomes can be really important for is sort of in the intake of things like cobalamin and folic acid, their ability to help facilitate the digestion of difficult to metabolize materials, the defense against specific pathogens. We can see them, certain communities of bacteria being associated with a risk of obesity later in life, for contributing to the development of diabetes, for evidence of a connection with arthritis and periodontal disease, for how well somebody might metabolize Tylenol, for how red meat is sort of broken down and how it might relate to cardiovascular problems. So there's a wide range of ways that we see, and there's a lot of sort of presentations out there too about people, promoting, sort of doing, looking at your own gut microbiome and then giving you recommendations on your diet. There's evidence to suggest some of these connections, but I'll just say that to me personally, it seems like we're still a bit away from really having a good, strong understanding of these relationships. But they are really promising, and there have been things to show that your gut may be signaling your brain, for instance, to like target eating more sugars in certain instances. And they might, be controlling your behavior. So maybe we're all just, slaves to our microbiomes. Who knows? But all is to say is that there's a lot of really interesting science here that we're still trying to get our heads around. Another really important area for the application of genomics is in the study of cancer. It's the leading cause of death in many countries. Cancer is described as basically cases where cells escape the normal controls of cell division. And this leads to an unrestricted cell proliferation and breakdown of cell adhesion mechanisms. And it's a disease really of the genome, right? Where the genome becomes unstable. And this instability then leads to the formation of things like tumors. And we have this case where we have this proliferation of these cells, these tumors forming, and we see significant differences between the genomics of a tumor and a normal tissue. And when I mean differences, I mean that their genetics are significantly different from each other. Their genomes are actually divergent from each other. And this is because mutations that initiate to allow for cancer to begin can then also continue to accumulate and cause other types of large-scale mutations like recombinations and crossover events that can accumulate within tumors and lead to very different genomes among the various cells that make up tumors. And so when we look at cancer, we're actually looking at a pretty heterogeneous population of cells often. The source of this genetic variation, of this heterogeneity, and the reason why we see such a success of these cells is that the main places where these mutations initially occur are in regions that regulate things like cell proliferation or related to required repair of DNA damage or control apoptosis, all things that allow, these mutations would allow these cells to escape the normal roadblocks in the cell cycle pathway, right? So getting past all these different gateway things that would sort of regulate cell growth, that would lead to apoptosis, that would control the accumulation of mutations. In the end, we end up with sort of two main types of genes that we think are associated with cancer. These are oncogenes, which is a gene that has the potential to promote cancer development, and tumor suppressors, which act to suppress the cell cycle and keep tumors from forming. These mutational patterns, though, are starting to become a lot better well known through the study of these tumor genomes, of these cancer genomes. And we're beginning to gain a lot more confidence, precision in using mutational patterns to both drive diagnosis, prognosis, and choice of treatment. So by looking at these genomes, we are starting to be better able to understand how best to give people advice on how to approach and consider risk in their lifestyles, in their choices, in their health, as well as being able to treat specific types of cancer. So what about SNPs? When we think about SNPs and cancers, we can see that there are definite mutations that people can inherit that would indicate various propensities for developing cancers. One of the most well-known cases of this is for the gene BRCA. And so for the BRCA gene, this BRCA gene, there are specific mutations that we can know that are well associated with an increased risk in the formation of cancer. And in this case, sequence analysis can help us predict the progression or outcome of a disease. It can identify a certain amount of risk involved in having that mutation. And it can also help us choose what is the optimal treatment. So if we see certain types of enzymes or different parts of the cell cycle being affected, maybe we can actually target different parts of the genome or different genetic elements to identify tumor cells or cancer cells versus regular cells. And tumor progression is known to involve lots of mutations and divergences that allow for the further divergence between tumor and normal cells, cancer and normal cells. Generally, we think of cancer following sort of a two-hit hypothesis framework, right, where we need 2 hits because you would likely have 2 copies of a specific gene, especially an autosomal gene, right? And you might say you were looking at a retinoblastoma gene, and we had an example of sporadic retinoblastoma, where you would start with 2 healthy copies. You then might have a mutation and lose a copy in a cell, but you'd have to have another mutational event and lose that second copy before the tumor would actually be able to form, right? Because you'd have to lose both copies of retinoblastoma in order for that to happen. In familial retinoblastoma, you might have inherited already one of these bad copies, and so you only have 1 good copy, and so you've already had that first hit, and so all you need to happen is to have the second hit, have a mutation occur in retinoblastoma that's not repaired, and you would get tumor formation. And so this is why people who inherit a bad copy of this gene have a higher risk of developing the disease versus someone who hasn't, because you have to have basically 2 events versus 1 for someone who hasn't had the disease versus someone who has a family history of such. If we think about, again, common tumor suppressors like BRCA1, BRCA2, or PALB2, we can see that when we think about BRCA1, we think about breast cancer, about 12% of women will develop breast cancer, about 1.4% will develop ovarian cancer. But if we think about mutations in BRCA1 and BRCA2, if we find specific mutations there, we can see that about 60% of women, actually it's between like 50 to 80% of women, you're at a higher risk, 50 to 80% increase of developing breast cancer, and also a much higher risk of developing ovarian cancer. Note that these aren't 100%, so just because you have a mutant or variant in this gene does not mean that it's absolutely guaranteed that this will happen, but your risks are significantly higher. So there can be a lot of benefits to knowing that. Along these lines, it's also interesting to note that different populations and origins of people have different types of mutations, and so Ashkenazi Jews have very specific mutations that are more common to people from that ethnic background versus, say, people from Denmark or China. So there are differences in which mutations we tend to see in people from different places. However, these cases do not account, from all the genes that we know about, they only account for a percentage of the cases, maybe around 25% of all cases. So there's a lot about breast cancer we still don't understand. So just because there are plenty of people who get breast cancer who don't have any of the known markers for it. And we're still learning about why that is. There could be environmental factors, there could be genetic factors that we don't understand, there could be a lot of developmental factors, there could be a lot of different factors that could be contributing to this process. One of the tools that can be used specifically in the study of the genomics of cancer is this process of whole genome sequencing. And this will basically is a way for us to get a good accounting of the entire set of SNPs that we see in cancer cells. And this includes novel and non-coding regions, help us see rearrangements, help us account for differences in copy number variant that we may see. And all of these things can be really important in thinking about, well, what are the real differences, mutational differences between these two groups, between healthy and non-healthy, or healthy and cancer cells, or even between different types of cancers. So for instance, if we're looking at something like diffuse large B-cell lymphoma, and we're comparing it to some non-hematologic cancers, so some non-blood cancers, we can see that there are certain, over here we have an amplification, we're talking about an increase in copy number variant. On the right, we're seeing a decrease in copy number variant. And what we actually see is that there are specific profiles that we see associated with large diffuse B-cell lymphoma that are going to be a little bit different in these cancers versus other types of cancers. So, for instance, we might see an increase in amplification of copy number variants along like chromosome 9 and 10, where we don't see that in non-hematological cancers. So we might see a decrease, we might see a lot less of an influence of deletion events in chromosome 10 than in other non-hematological cancers. Right, so we can see some of these variants, and actually by learning this, we can look to identify, what are the specific genetic types or genotypes associated with certain types of cancer. So insertion deletions are important, and often rearrangements are really important. So if we look at genes like the BCR gene on chromosome 22 and the ABL gene on chromosome 9, sometimes they'll recombine to form something called a Philadelphia chromosome, where they've been some crossover events here, some rearrangement, and basically this will form a BCR ABL1 oncogene. And when the formation of this oncogene can lead to the occurrence of chronic myeloid leukemia. So these sorts of mutational events can be really important. And it's only through genome sequencing that we're going to observe them. It's not just genomics, though. Transcriptomics is also really important. When we look at the epigenetics of cancer, we can see big changes in base methylation between different types of cancers, between cancer and normal cells. And we also see huge changes in gene regulation in terms of both the genes that are being expressed and also the expression of microRNAs and non-coding RNAs. In fact, you can use, this is an example from a cancer transcription atlas produced by UCSC. And what this shows is that there's a whole bunch of different types of cancers that have very specific types of gene expression profiles, so much so that we can cluster them based upon what their gene expression profiles are. So we can actually identify what are the genes that are active and important, and how they might differ among these different types of cancers. And by understanding what genes are important, that can also help us identify different targets that may be important in fighting those cancers. So using something like temozolomide, which is a treatment for glioblastoma, which causes methylation with methylase poly-G tracks, right, and can help sort of inhibit activity of cancer cells. And there are some other things that we can do, like targeting small RNAs involved in hepatocellular carcinomas in order to suppress tumorigenesis. So there are like targets that we can say, oh yeah, look, we see this specific thing active in these cells. We can then look at that and say, this is how it's different. Let's target this thing that's different, and maybe that can be a helpful way of looking for treatment targets. Last thing I'm going to talk about is just sort of, okay, we've got this approach, we've got all these tools now we can use for both personalized and precision medicine, but there is a big movement to sort of really rely on these genomic resources in a lot of different ways, and one of those ways is to actually give access to you and me as regular citizens to our genomic data. And so this decrease in cost of sequencing has led to increased access to genomic technology even for us. And so the goal is, oh yeah, let's sequence a human genome for $1,000 or less. And guess what? As a company, as an organization, we'll use that information to tell you what it means. So there's a whole suite of direct-to-consumer sequencing platforms out there. Probably the best known are things like 23andMe and Ancestry.com. But then there are other ones like Veritas and Nebula Genomics, which will actually give you whole genome sequence. There are screening and more targeted clinical tools like Myriad Genetics and Color and Family Tree DNA. These things will help sort of target your DNA targets and also give you some genetic counseling support. Sometimes with specific things like doing genetic screens before you might consider having children to try to figure out what you might be concerned about or worried about. And I think one of the main things here as we look at these direct-to-consumer platforms is understanding that there is actually, some of these are chip-based. So they're using microarrays like 23andMe and Ancestry.com, and some of them are actually going to be more sequencing-based, things like Veritas and Nebula, right? And so there's a big difference between the kind of information that you're going to get on a chip versus whole genome sequencing, as the chip is going to be charged for specific SNPs, whereas whole genome sequencing will give you information across a whole wide range of different types of genetic variation, copy number variants, rearrangements, things like that, things of that sort. If we look in at some of these, and maybe I'll show you a little bit of this in class, but if we look at something like 23andMe, which is probably the most commonly used one, right, costs about $200. And by the way, the prices here, I don't know if they're accurate or not. I did my best. I think if you want to go Black Friday, they have big discounts. But I'll just give you a rough ballpark. But if we look at something like 23andMe, right, this is a microarray based approach. It's got over sort of 600,000. I think it's closer to 690,000 SNP markers now. And you get reports on all kinds of things related to ancestry and your health. And a lot of this health stuff, I would say take it or leave it. There are some things that could be helpful there, but again, they are all indicative of risk factors, which is not sort of manifest destiny. It's not destiny, excuse me. That's definitely going to happen, but it may be something that targets something you want to be aware of. That's something you want to track. That's something you want to keep your eye on. There are also some really interesting parts of ancestry. So, I've used it to find some interesting things about my own ancestry. You can use it to find relatives. I can talk in class about that too. I've been part of some efforts where that's been used in a legal context. So there's a lot of really interesting data out there. I will sort of offer a caution though, is that, you're putting your genomic information in the hands of a company. So you do want to be a little bit careful and informed about what you're agreeing to and what you're comfortable with. Who's going to have that information in a world in which, maybe all companies are eventually going to get hacked at some point. We do want to be aware that, putting out our genomic information may or may not be to our benefit one way or the other. So, but there are some really interesting ways that this may sort of empower us to be able to take more control of our own health, to better advocate for ourselves, to better communicate with our doctors. So there's a lot of really interesting outcomes that could come out of it. All right, that's my lecture on human health and genomics. Hopefully we'll be able to come back and chat about these things in class. And thanks for your attention.